Plants have evolved physical and chemical mechanisms to protect themselves from infection by pathogens such as viruses, bacteria, fungi, worms, and even parasitic plants. The chemicals produced by plants as part of their defense affect the behavior and physiology of animals that try to consume them. This includes the physiology of humans. The chemicals that plants produce as a defense have been used by humans as sources of medicine, with nearly 30% of all medicines based on chemicals that plants produce to defend themselves. The epidermis of plants represents the first line of defense from pathogens. The cells of the epidermis have thick cell walls and are covered in a waxy cuticle. Viruses and bacteria are not able, on their own, to penetrate through this covering. However, as we mentioned in a previous lecture, the presence of the cuticle also necessitates the presence of stomata, so that CO2 can diffuse into the leaves. These stomata can serve as entry points for bacteria, protists, and fungi to gain access to the interior of the leaf. For example, here on the slide, we have an example of Phytophthora infestans growing through the stomata to access the interior of this leaf. Phytophthora infestans is an oomycete, a protist, that caused the Irish potato famine. Some bacteria and fungi can secrete a chemical that prevents stomata from closing, increasing their chances of infection. Pathogens can also gain entry and bypass the walls and cuticle through wounds or via the piercing mouthparts of insects and nematodes. Some pathogens are able to weaken the epidermal cell walls by secreting enzymes that degrade the cell wall. These pathogens are then able to force their way into the plant's interior. Some pathogens move cell to cell, once they're inside the plant, by secreting cell wall degrading enzymes. Some fungi and bacteria take this route. Viruses are able to use the plasmodesmata, the connections in the plasma membrane between cells, to move from cell to cell. For longer distances, pathogens are able to use the vascular system of the plant as a highway. Viruses and some bacteria are transported by the phloem, and other bacteria and fungi are transported by the xylem. Pathogens can be broken down into two categories: biotrophic or necrotrophic. Biotrophic pathogens obtain resources from living cells, and the majority of this type of pathogen, with the exception of viruses, do not directly enter the host cells. Instead, biotrophic pathogens release chemicals that trigger the host cells to secrete compounds that the pathogen can consume. Viruses require a living host, but they enter the host cells and hijack cellular machinery in order to replicate and thus reproduce. Necrotrophic pathogens, on the other hand, obtain their nutrients from dead cells. These pathogens will kill cells via toxins before feeding on the compounds that result or become available after cell death. Bacteria and fungal pathogens can fall into either camp, depending on the species. They are either biotrophic or necrotrophic. There are also plants that serve as pathogens of other plants. These are parasitic plants that infect other plants in order to obtain nutrients or other resources. Parasitism in plants has evolved multiple times, and there are currently over 4,000 known species of plant parasites. A common mode of infection used by these plants is to penetrate the stems or roots of other plants using a specialized structure and then tap into the host plant's vascular system. Much of the greenery on this tree is actually mistletoe. This parasitic plant is able to photosynthesize itself, but it connects to its host plant's xylem to obtain water and nutrients. Because it takes advantage of its host like this, mistletoe does not have to produce roots. Rafflesia produces the world's largest flower, but there's very little else to this plant that is visible. The rest of the plant body is found entirely within the vines that make up its host, and it obtains everything it needs from its host. This last parasite is called cuscuta, or daughter. This plant penetrates the stems of many plants, and like the Rafflesia, it receives everything it needs from its host. A plant's immune system allows it to detect and respond to pathogens. The plant species that can be infected by a specific pathogen are called the host plants of that pathogen. However, not all infections have a significant effect on the host plant. Pathogens that can overcome a host plant's defenses and result in disease are termed virulent pathogens. Avirulent pathogens, on the other hand, result in damage to only a small portion of the host plant, and the majority of the pathogen's infection is contained. It is the genotypes of the pathogen in the host plant that determine whether the pathogen will be virulent or avirulent. The immune system of a plant has two components, basal resistance and specific resistance. Both of these components rely on protein receptors that recognize pathogens as foreign by binding to molecules released by the pathogen. The binding of the molecules to the protein receptor then triggers a series of changes and reactions within the plant that allow it to resist infection. What varies between the two components of the plant's immune system is the location of the protein receptors. The basal branch of the plant immune system consists of receptors located on the plasma membrane. These receptors recognize molecules like flagellin or chitin generated by a broad class of pathogens. These molecules are highly conserved, meaning that they have remained relatively unchanged between species and over evolutionary time. When these molecules bind to basal resistance receptor proteins, they trigger an array of defense mechanisms that serve to prevent infection. However, some pathogens have evolved an ability to overcome these defenses raised by basal resistance. These pathogens have overcome basal resistance by producing AVR proteins, which enter into plant cells and facilitate infection. From here, these proteins can block the basal resistance response or trigger the cell to produce molecules that the pathogen requires for growth. This is where the second branch or component of the plant immune system comes in. This component is called specific resistance, and it targets these AVR proteins. The receptors employed by specific resistance are found within the cell and are called R proteins. The R here stands for resistance. Each plant has hundreds of these protein receptors, which are encoded by genes collectively called R genes. Each R protein recognizes a specific AVR protein and binds to it. This binding then blocks the effect of the AVR protein and triggers additional defenses by the host plant. This component of the plant's immune system results in an evolutionary arms race between new AVR proteins produced by the pathogen and the evolution of new R genes by the plant. Because this branch of the immune system depends on interactions between specific plant and pathogen genes, it is commonly called the gene-for-gene model of plant immunity. Once a pathogen has been detected, plants can employ several methods to protect themselves. Plants can reinforce their natural barriers, such as strengthening their cell walls, closing stomata, and plugging their xylem, which can impede the spread of the pathogen. Some plants will produce antimicrobial compounds to counteract the bacteria or fungal pathogen. A third type of response is called the hypersensitive response. In this type of response, the cells that surround the site of infection trigger the reinforcement of cell walls and cell death by producing reactive oxygen species. This creates a barrier of dead tissue around the infection. This dead tissue isolates and halts the spread of biotrophic pathogens. This photo shows a tobacco leaf that has been infected by tobacco mosaic virus. A hypersensitive response then results in necrotic zones surrounding the initial site of infection. This prevents the spreading of the virus to other parts of the plant. While animals can isolate infections to some extent, plants are able to do this to a much greater extent. For example, they can plug parts of their vascular system to prevent the spread of pathogen. But this is not always successful and can result in harm to the plant and contribute to its death if the pathogen evades containment. There are a group of virulent pathogens that traverse through the xylem and cause a collection of disorders called vascular Wilt disease. These pathogens can kill their host if the plant's defense block water transport to the leaves. Here on the left, we have a case of Panama disease. This is caused by a fungal pathogen that infects bananas. On the right are grapes that are affected by Pierce's disease, caused by a pathogenic bacterium. Plants can acquire immunity to pathogens after being exposed to the pathogen in another part of the plant. This form of immunity, called systematic acquired resistance or SAR, allows them to resist further attack by the same pathogen. Systematic acquired resistance was demonstrated in an experiment carried out by plant pathologist A.F. Ross. He infected a leaf from a tobacco plant with the tobacco mosaic virus, or TMV. The infected leaf became mottled looking as it developed dead patches characteristic of the hypersensitive response. Then Ross infected another leaf on the same plant a week later. This second leaf displayed only minor signs of TMV infection. The reduced response to the second infection indicates that a signal had been transmitted from the originally infected leaf to the undamaged parts of the plant, and that this transmitted signal triggered the development of an immune response to protect the plant from further infection. The signal itself is still unknown, though experiments have shown that salicylic acid is required for SAR. Since these classic experiments, we've learned that, in general, SAR is not highly specific. It happens in response to pathogens that result in dead plant tissue, caused by a hypersensitive response or by necrotrophic pathogens. SAR usually activates defenses effective against broad classes of pathogens. This differs when it comes to viral infections. As we'll see on the next slide, in these cases, plants generate a response that is targeted to a specific pathogen. Viruses can be extremely harmful pathogens, and in reaction, plants have evolved multiple responses to viral infections. There is the hypersensitive response, which we've already discussed, and a more specific targeted response, which we'll cover now. The targeted response takes advantage of the fact that most plant viruses have genomes made of single-stranded RNA molecules, or ssRNA. This ssRNA must be replicated into double-stranded RNA, or dsRNA, during the infection by the virus. However, plants do not normally make dsRNA, and so this marks the viral RNA as foreign. And so here's a look at the plant's targeted defense against viruses. It starts with a virus infecting a cell by injecting its ssRNA genome into the cell. This is then replicated into dsRNA. The viral dsRNA are recognized by the plant cells as foreign, and the plant cell produces enzymes that cut these dsRNA molecules into small chunks or sequences about 20 to 25 nucleotides long. These fragments are called small interfering RNA, or siRNA. Protein complexes within the cell then bind to the siRNA and use the siRNA nucleotide sequences as a targeting system to find the viral ssRNA by looking for sequences that are complementary to the sequences found in the siRNA. Since the siRNA is produced from the dsRNA that is replicated from the ssRNA, the small interfering RNA will match the ssRNA, and the protein complexes can then destroy the viral ssRNA once a match is made. The siRNA can then also function in acquired immunity. Once they have been produced in one cell, they can be transported via plasmodesmata to neighboring cells, and to the phloem where they can spread to the rest of the plant, permitting other cells to target the viral ssRNA. Well, on the previous slide we covered an example of the plants targeting a pathogen's genome. Some pathogens target the plant's genome during infection and alter the host plant's biology. There are some pathogenic bacteria that can insert their own genes into the plant's genome. The plant then transcribes and translates the inserted genetic material, which results in products that alter the plant's physiology in response to the pathogen. One such pathogenic bacteria is Rhizobium radiobacter, which used to be known as Agrobacterium tumefaciens, or agro for short. This is a bacterium found in the soil, whose virulent strands infect the roots and stems of many different plants. Wounds typically serve as entry points for Rhizobium radiobacter into the plant. An infection by this bacterium is called crown gall disease due to the tumor or gall that forms at the site of infection. Rhizobium radiobacter alters the growth and metabolism of infected cells by inserting some of its own genes into the chromosomal DNA of the host plant. Virulent strands of Rhizobium radiobacter contain a plasmid called the Ti plasmid. The Ti plasmid is a small circular molecule of DNA that contains the genes which are destined to be integrated into the host cell's genome, as well as all the genes needed to make this transfer of genetic material. Some of the transferred genes cause the infected cells to proliferate and form the gall by inducing the synthesis of the two plant hormones auxin and cytokinin. These gall cells are then triggered by other transferred genes to produce specialized compounds that the bacteria can use as sources of carbon and nitrogen for growth. The bacterium's ability to incorporate its DNA into the genome of its host provides a powerful tool for biotechnologists to engineer plants. By replacing the genes on the TI plasmid that are responsible for gall formation with genes of interest that potentially may deal with nutrition or disease resistance, biologists are able to improve crops and other plants. If you have any questions, please let me know via the Q&A discussion board.